Okay, we resume our discussion on signaling, focusing on the mechanism of information transfer. On this slide, we see the two primary molecular mechanisms whereby the information is transferred between proteins. And the dominant one will be this phosphorylation event. And that is, there are two types of proteins, protein kinases, which put phosphoryl groups onto proteins, and phosphatases, which cleave them off. When you put a phosphate group onto a protein, this phosphate has negative charges that will alter the electrostatic balance of the protein, can likely lead to a conformational change and a change in activity, and now this is an active protein. Many signaling proteins are kinases themselves, so a kinase will activate this protein and then it will in turn activate other proteins. Phosphatases are the opposite, and they will cleave this phosphate off, allowing the conformation to return to the inactive form. And although this is generally the way biologists think of it, that the phosphorylated form is active, that is completely arbitrary. It is completely modular. A phosphorylated protein can be turned off and the dephosphorylated one can be turned on, or vice versa. But again, this is the way that biologists normally think of it. Completely analogous but slightly different is proteins that bind GTP or GDP. Again, I think it's clear to see that the difference between this state and this state is a single phosphate group. So there's a charge difference between these proteins, conformational changes, and activity changes. Now the difference is here, the ATP is being hydrolyzed and the phosphoryl group is being covalently attached to the protein. In this case, this protein binds the molecule GDP or GTP, and the way it'll occur is this GTP would get hydrolyzed, the phosphate group would come off, the GDP molecule is still being bound. Eventually, this GDP molecule will diffuse out of the binding site for it, and a molecule of GTP will diffuse in. But it's the same relationship here in terms of the difference of one phosphate group, whether covalently bound or binding non-covalently, that will change the activity and the relay capability of this signaling protein intermediate. The last critical thing to mention is when we talk about a protein getting phosphorylated, it's one of three amino acid residues: serine, threonine, or tyrosine. This slide examines in a little bit more detail scaffolding proteins. They have two primary functions. One is holding signaling intermediate proteins in close contact so that when a signaling molecule binds, these proteins are activated. You don't have to wait for diffusion. The relays can occur very quickly, and so you have a speed and efficiency associated when a scaffolded protein is organizing signaling protein intermediates in the correct order, close to one another, so that a signal may be transported very quickly. A second function of scaffolding proteins is to minimize crosstalk between different signaling paths. We've already seen some examples where signaling pathways will share some components. We'll see one on the next slide. And so the question is, how come if a signal comes in perhaps at this receptor, how do you keep the signal being transmitted here and not perhaps crossing over and proceeding down another pathway? By holding the proteins close to one another so that they can't get mixed up, you can maintain the fidelity of a signal along a specific pathway. And here's an example where there are two different signaling pathways used in yeast, where one promotes mating, one senses high osmolarity, and they both use the same protein, kinase A, as a signaling intermediate. To make it clear is, what if the osmolarity sensing receptor was triggered? It would activate kinase A. You do not want to have this trigger a mating response. What you want to do is synthesize glycerol as an osmolyte to increase its internal osmolarity. So again, the fidelity of the signal going through the shared kinase A can be maintained because scaffold 1 holds kinase A next to kinase B. The last thing to mention on this slide, which we'll see in subsequent slides too, is these kinase intermediates called MAP kinases, which is MAP stands for mitogen activated protein kinase. And once again, biologists have a wonderful naming system. The kinase that phosphorylates MAP kinase is the MAP kinase, and so on. And there's even a few examples of the word kinase being used 4 times. But this general MAPK sequence, and the prototypical one is with 3 MAP kinases, is a shared set of proteins that is used over and over again evolutionarily to conduct signaling pathways and transmit information about the cellular environment. Because kinases are enzymes, they can amplify signals, right? One activated protein could phosphorylate 10 or 100 downstream proteins. In this way, you're able to have a single ligand molecule bind a single receptor and yet phosphorylate thousands or millions of proteins, copies of the proteins, so you can have a large quantitative response downstream. One of the best quantitative examples of signal amplification occurs in rod cells in the eye, in which a protein called rhodopsin will absorb a photon of light. This will activate a G protein, a single photon activates about 500 G proteins. These G proteins will eventually hydrolyze approximately 10 to the 5th cyclic GMP molecules. Those will close cation channels, and a difference of about 10 to the 7th sodium ions are prevented from translocating through the channel. This produces a membrane potential change of about 1 millivolt, and that triggers the neuronal impulse to your visual cortex in the back of your brain. So in this case, you have an amplification of perhaps 10 to the 7th from 1 photon influencing the movement of 10 to the 7th sodium ions. It's amazing when you think about the sensitivity of the eye. All this can occur. We can see a single photon. We miss about 2 out of every 3 photons. If you fire photons one at a time at the eye, approximately every 3rd photon you will produce a signal. The other times the photon will travel in between the cells and not get absorbed by one of the rhodopsin molecules that are present in the rod cell. I've always found it amazing that the light has to traverse the cellular body to finally get to the sensing part. You might have thought that it would make sense to have the photons enter from this side so that they can't get diffracted by the cell body and miss sensing part, but obviously nature has got that worked out pretty well. Okay, thanks for listening.